All right, just a quick overview to wrap up what I did really fast today if you need it. Just going to go through one replication fork and all the enzymes in the order. So first take note, this is a replication fork. Notice the 3' 5' anti-parallel 5' 3'. You should be able to look at this first of all and tell me which side's going to be the leading strand and which is going to be the lagging strand. So if you want to pause and think about that for a second, but I'm just going to kind of move on. And remember, we always read DNA 3 to 5'. So those are both my template strands. So this strand is going to be read in that direction, and this strand on the left is going to be read kind of down. And if you think about, let's start out with, I want to kind of color code this. So number one, I said, remember, was helicase. So let's say we have helicase right here that has unzipped it and is going to continue up there unzipping it. Okay, so I'm not going to write what they do because it's all written in the notes. I just kind of want to show you. But helicase, if you want to write, remember, don't say unzips it, but it basically opens up the replication fork. It breaks the hydrogen bonds. Then the SSBs come in, the single-stranded binding proteins. And those aren't really an enzyme, but I'll just draw a few. And they are just on the outside here, and they kind of hold the strand open so that these base pairs don't reanneal. Because I'm not drawing the bases on here, but remember there's like an A and a T and an A and a G that pairs up with a T and an A and a T, whatever the base pairs are. So, you know, those are attracted to, these base pairs want to go back together and reanneal. So the SSBs keep it from reannealing. I'm going to erase those bases just to simplify my drawing. Then the third enzyme that comes in, you'd think it would be DNA polymerase to just, once we've opened it, we just now copy it. But remember what we talked about in class today, DNA polymerase, the DNA polymerase that we're going to use, can't bind on to single-stranded DNA. So it can't bind on to these and start reading them. So we're going to have this enzyme primase, I just call it primase, which is going to add the RNA primer, which is made of RNA nucleotides that are going to base pair with my DNA. And let's think about where those would be added. Now start thinking about the leading versus the lagging strand. So if you picked the leading strand on the right, this is going to be my leading strand. So the primer is going to attach down here. And those are RNA nucleotides. If I looked up close, you know, like here, and I had like an A and a T and a G and a C, then the RNA nucleotides that were paired with it, remember U goes with A still goes with T, C, G. So this is like my RNA primer. I'm just not drawing the base pairs, okay? But these RNA nucleotides would have ribose in the backbone, and we're going to have U going with A. Remember the anti-parallel nature. If this is the 3' end of my DNA, my template, then that is the 5' end, okay, of my primer. So the primer, that would be the 5' end of my RNA primer, and this would be the 3' end right there. So it's always anti-parallel. Okay, and then remember at the 3' end, there's what we call the 3' hydroxyl group kind of hanging off there. That's what the DNA polymerase can extend. And up here, my primer on the other side is going to be up here because it's going to be synthesizing DNA in this direction, right? Because we always read 3 to 5, the template. So this would be the this would be the 5' end of the primer, and this would be the 3' hydroxyl group hanging off of my primer, because the primer is going to be anti-parallel also to the DNA. Everything's always anti-parallel. So that's the primase. Remember, now that that's there, my DNA polymerase, I'll make this 4, my DNA polymerase 3, polymerase, whatever, DNA polymerase 3. Now it can bind on because what it requires, it needs double-stranded nucleic acid to bind on, And we now have that here. It's DNA, RNA double strand, but we now have double stranded. And it can extend the 3' end of a strand that's already started. It extends the 3' end at the hydroxyl group. It extends the 3' hydroxyl. So it's going to see this 3' hydroxyl and this double strand, and it's going to hook on here. Okay, so that's my DNA polymerase 3. And it's going to start adding DNA nucleotides and start going in this direction. DNA synthesis is going to be in the 5' to 3' direction. But we still have, now we have this strand that's kind of like a little bit of RNA nucleotides, and these are DNA nucleotides. And this is going to be my leading strand, continuous, just following the helicase. That's the only primer I will need. And over here, we're going to have the same thing. We're going to have my DNA polymerase 3 can now hook on because I have double-stranded nucleic acid. I have a 3' hydroxyl that it can extend. And so it'll then it'll move down this direction and synthesize the new strand again. This will be anti-parallel. Okay. So that's initially. So both sides we might, like it just started to unzip. I'm going to have a primer here on the leading strand because it's going that way. And I'm going to have a primer here on the lagging strand and it's going this way. Now, this helicase is going to continue to unzip. So this picture down here, I say later, okay? So here's, let's say this was my, this is the original, let's call this the first primer on my leading. That's my first primer. This is my leading strand. So my leading strand only needs one primer. This is my lagging strand on this side. We're going to see, initially it needs one primer, but we're going to see it's going to get more. So, and let's call this the, this is the first primer on the lagging strand. These are both the first primers. And I'm going to label these down here. This is still the first primer that we initially put on there. And my DNA, like I still have my helicase here. It's still continuing to go up. And my DNA poly 3 might be to here, but it just keeps going. Okay, I don't have any more primers on that side. Okay, over here, let's say this was my initial first primer that I drew up there on the other picture. And my DNA poly 3 initially bounded on here and is now down here at the bottom, okay? But now do you see, because this has been unzipped further, so this was the first primer on the lagging strand. Now, because the synthesis of the new strand, which is shown here in green and red, is going this way, but as helicase goes this way, I now need to jump back and go, oh crap, I need to do another primer. Okay, so primase will come in again and it'll add, we'll call this the second primer of the lagging strand. And then DNA polymerase will hook on to that and extend that. Okay, and so on. I could keep drawing this unzipping higher and higher. And the lagging strand, I'm going to have to keep jumping back, adding another primer, and then synthesize the new strand this way. Whereas my leading strand can just keep going. I don't need any more primers, okay? So now what's the problem? Well, I have two problems now. I have my new daughter strand, which is shown in green and red here, has RNA nucleotides in it, and I don't want that, so we're going to chew that out and replace it with DNA. So let's do that as what number was I on? That was 4. So number 5, we're going to have RNase H, okay, and DNA polymerase, I'll call it DNA poly 1. Okay, these are two different enzymes. Remember, what RNase H does is it removes the RNA nucleotides. And then the DNA poly 1 is going to replace or add DNA nucleotides back into that space. And these two enzymes are sort of in picture like a like an enzyme complex. I kind of talked about this today in one of my classes. It's a blob where maybe there's like an active site here. Maybe the RNase H active site is on that side, and maybe the DNA polymerase 1 active site is on that side. So think of this like there's like two enzymes that are connected, and this wheel, this is an enzyme that's just going to turn. So it's going to come through here, and the RNase H is going to chew out the primer and replace it with DNA. So I don't know how to draw this, but I'm going to draw this as just sort of okay, so this is the RNase H DNA poly 1 complex. And what it's going to do is chew out the RNA nucleotides and replace that, I'll do DNA in green, with DNA nucleotides. There's still going to be a little break right there, what I call a nick in the sugar phosphate backbone. So we are going to need ligase in this one place. And the same thing's going to happen on my lagging strand, okay? So this is going to be it's going to happen here and here. Now it's getting kind of complicated, but it's going to chew out my RNA primer, and I don't want to erase everything, and make DNA. Okay, let's try to get all the RNA out of there and replace that with DNA. Okay, so that's what RNase H and DNA poly 1 is. And then the last thing that's going to happen is 6 is the enzyme DNA ligase, or sometimes we just call it ligase. Think of this like the glue. And what this is going to do is create a phosphodiester bond Hi.
Check out the nest?
Sure. It's going to create a phosphodiester bond between the sugar and the phosphate in the backbone. Okay. So I like to say, like you see here, we sort of have this little break right here. We have there's bases, right? The A's and T's and G's and C's are all along here. And we have a sugar phosphate backbone. But right here, there's like what I call a nick. I sometimes call it a nick or a break in the sugar phosphate backbone. And that's where DNA ligase is going to come in. So DNA ligase is going to come in and basically create that bond. And it's going to do that here between all these Okazaki fragments and this one place over here that we had primer on the lagging strand. And after that, once everything comes off, then we should have a new strand of DNA that's continuous. The Okazaki fragments will be all glued together. There's no RNA in it. And same thing over here. And it'll just keep going. All right. Don't worry about topoisomerase. I'll talk about that one later, but that should help tonight, hopefully. A couple things I just wanted to go over in the notes real quick. I did that in class today, and I did this in class today. You might want to just jot this down. I didn't get to this picture. Kind of same thing I just kind of talked about. Again, here's my helicase unzipping. Here's my leading strand with a single primer and then new synthesis. I had this primer, that synthesis. This is the second primer, that, second primer, that, and it'll keep going. And this shows you later, see how now there's 4 Okazaki fragments as I keep unzipping. But just some notes here you might want to jot down. This is my lagging strand, which is going to have I guess I should put here, it has Okazaki fragments. There's no Okazaki fragments on the leading strand. So these are the Okazaki fragments. So the lagging strand has Okazaki fragments. The DNA synthesis goes in the opposite direction as helicase. So notice helicase here in purple is going to the left, whereas my DNA polymerase 3 that's synthesizing DNA is going to the right. And that's why it does this, has to keep jumping back, synthesize that way, jump back, synthesize that way. Or we use this, you'll see this word in the homework and on quizzes. DNA synthesis is discontinuous on the lagging strand because we have to keep jumping back and starting over. And it's going to need many primers. Whereas the leading strand is the opposite. You only need one primer just to get it started. You'll notice that the synthesis of DNA is going in the same direction. It's just following the helicase. And we say this is the synthesis of DNA is continuous. You'll just see those words, so you might want to just jot those down. You'll see these words a lot in the homework and in pop quizzes: continuous, discontinuous. This picture just shows you the whole thing again, sort of in a different picture. Again, here's my origin of replication, where we're going to open it into a bubble. This should be DNA polymerase 3, this yellow enzyme. Notice how there's, so let's just look at one replicate. Here's my DNA helicase. My SSBs are holding it open. Let's just look at this replicate. So I put a square around this. Let's just look at the right side, this replication fork. And then down here, you can see sort of a time lapse as you go down. There is a helicase here in brown. I drew it. That's continuing to zip. It's still here. It's continuing to unzip. It's here, whatever. They just stopped putting it in the picture, but it's there. And so you can see initially, here's my lagging strand. Starts out with just one, and my leading strand with one. But as we jump down here, notice now my lagging strand has two. This was the first one. So this one, this strand, and this strand are the same one. We just unzipped further. Then I had to come back and start over. And then here you can see, so that strand, this strand here is the same, that original first strand. And you can see here the primer has been chewed out and replaced with DNA, and DNA ligase is sealing that together. And just a note, if you're studying from this picture, the picture is missing. It doesn't say anything about the RNase H and DNA poly 1 that come in and chew out the primer and replace it with DNA. So this picture just doesn't show that, but if you're studying, just add that in there. And I think the rest we talked about. So that's it. Good luck.